Welcome to this Cell and Gene Therapy Insights Fast Facts presentation with C Repligen Company. We're joined by Paul Mania, who's here to tell us about slope spectroscopy in gene therapy applications. Paul is a bioanalytics applications specialist who has been with C Technologies since 2017. Leveraging his studies in biological science from Duquesne University and industry experience as a laboratory technician, he now helps customers with method validation and implementation, getting solutions integrated and functioning as quickly as possible. If you'd like to ask Paul a question about his presentation, simply use the Ask a Question box to the right of the viewing pane. And with that, I'll hand over to Paul for today's presentation.
Hello everyone, I'm Paul Mania. I'm a bioanalytics application specialist for the SoloVPE and slope spectroscopy technology. I've been in the field for over 3 years. I've started in a clinical lab, so I certainly have some lab experience that carried over into instrument qualifications of the SoloVPE. So naturally now I've transitioned into bioanalytics applications and webinars. So what we're going to be discussing today is one of the applications for the SoloVPE, which is all revolved around gene therapy products and their processes. As mentioned, this presentation is going to focus on gene therapy applications for slope spectroscopy. Mainly, we'll be talking about the SoloVPE, but all of this does apply to our other instrument called the FlowVPE, which is the inline version. So I'll get right into here with the agenda. So we're going to be covering some gene therapy applications first and how slope spectroscopy can be utilized for their techniques. And the first thing we're going to do is talk about some of the challenges with the current gene therapy analytical solutions. And so after that, we're going to get into some fundamentals of slope spectroscopy. I think most would agree that understanding exactly how the technology works is going to help provide some context. So it's going to be clear later how it can be applied for gene therapy itself. And so after that, we're going to get into some examples and applications that can be leveraged using the SoloVPE. And lastly, we'll talk about customer success and why we believe we are different in how we handle our customer success, because throughout the entire process of SoloVPE purchase, we are actually with you along the entire way. And I'll certainly be explaining more of that at the end. So I'm sure everyone knows that this is a timely and burdensome process with gene therapy. There's many challenges associated with that. I think everyone agrees that we all want this process automated on a standard platform, easy to use, and preferably single use. So the context of this slide here is to talk about the issues with the current process, what we plan to do about it. The main goal here is speed to market, right? So this is certainly critical for product success and can actually affect production due to the timely nature of this process. So this slide highlights some of the main goals in some of the departments for gene therapy manufacturing. So PD, MSA&T, they're really focused on improving manufacturing of gene therapies, developing consistent and scalable platforms, which thus would increase manufacturing yields. Manufacturing QC, same idea. They want to reduce the cost of goods by improving therapeutic efficiency. So their goal is to employ analytics that will enable process optimization. So something important for this is to ensure that the innovative improvements are removing variables and failure modes. For AD analytical development, they're trying to achieve a more robust process, and all of this stems from the current lack of rapid analytical methods. So all in all, the main issue with the current gene therapy process is a high range of tolerance for error and the timely process that it has. So these are the current methods that are being used for gene therapy. I'm sure most people are familiar with at least one of these. And all of these have one thing in common, in that there's a large error tolerance, and all of them are time consuming in nature. And the time consuming process can result in extra expenses, especially when a result forces manufacturing to idle. So this certainly can affect batch amount, yield, and/or efficiency when you're forced to send your sample out for analysis and manufacturing is just waiting for a result. So this slide highlights the different techniques that are currently being used, and it shows the magnitude of the accuracy, validation, ease, and the expertise required. So as you can see, we have green plus signs, which are positive. These are good. So the more plus signs, the more magnitude of the category. So we can actually see SoloVPE has plus signs in each category because when we use SoloVPE, we just measure the sample raw. We don't have any other processes intervening, so we actually have a lot less variables to consider. So we're not actually sacrificing accuracy for expertise or vice versa. So I think this is a good time to get into slope spectroscopy method and talk about exactly how slope spectroscopy works and how we're different from UV and how we believe we're much better than UV and a drastic improvement. So before we get started on exactly what the technology does, this is the ASTM International E169 standard. It's the standard for good practices of techniques of uv-vis. So every 5 years, this document's revised, and most recently in 2019, the revision was made based on a wider adoption of slope spectroscopy in the marketplace. So they now include the adjustment of path length in the verbiage of this document here, and I'll explain a little bit of the adjustment of path length in just a moment. But this essentially is related to slope spectroscopy only on the SoloVPE and FlowVPE, but slope spectroscopy only, meaning that we're the only ones that perform this technique. So we've had a lot of client-supported data and posters coming out over the years, but to actually be written into regulatory standard now for uv-vis is very significant for us. And this should certainly make any of you who would be looking to adopt the Solo technology for the first time understand that you would not be the first person to take this on. It's actually been widely accepted for over a decade now, and certainly having backing of regulatory can only help. So the main issue with the non-solo VPE, traditional UV techniques is the dilution problem. So every single time we dilute, there is a variable associated with that. And it really depends on the dilution technique, how accurate it can be, but no dilution is perfect. So dilution will be associated with error and a time-consuming process. And you need to train your technicians on exactly how to dilute and ensure that they are all doing this very similarly. And so the real issue with the current technique is that you really never know how linear you are within the Beer's law equation, only because you're given a single absorbance reading. And so we can take a look at the traditional UV equation over here. It's exactly the same as the slope spectroscopy equation, except that we have our path length locked. And that's because we have to put our sample in a cuvette and we can't move this cuvette around. So this actually forces us to unlock the concentration and dilute it down so it becomes within an acceptable absorbance range so that we can ensure that our results are in fact viable and linear. With SoloVPE, we've actually redirected the light to come through the top. So now we fill a sample vessel to a certain height, and we can actually adjust how close this light is getting to the detector. So the distance from the tip of the fibrette, which is the fiber optic use and throw consumable that goes in for the samples, and the distance between the tip of this and the bottom of the detector is going to be our path length. So we can actually alter how close we are to this and lower our absorbance by getting as close to the detector as we want within 5 microns. So now we've actually unlocked the path length, which allows us to lock in our concentration. So essentially what we're going to be looking for is the change in absorbance over the change in path length, which is going to be able to give us the equivalent of an OD reading on the traditional UV. So rather than scanning across the sample compartment, we're now coming through the top. So all of that process actually results in this 10 data points coming down, which will create a slope regression, and every single slope regression is going to be associated with an R-squared value. So the system will lower to the 0 path length, find one absorbance value. So it is intuitive with the concentration, and it will change the path lengths per the concentration. It collects 10 data points. All of this is done within a minute. It actually takes quite a lot more time to explain the process than it actually takes. And then you're resulted with this. You'll get a slope result. And if you have an extinction coefficient, you will have a concentration result. And as I was saying, each result has an R² value for linearity. So that needs to be 0.999 or higher to be an acceptable value. So essentially what we've done is we've really just taken this path length variable in the Beer's law equation and we've put it underneath the absorbance. So we're going to look for the change in absorbance over the change in path length, which is now our slope. And again, the slope is going to be the equivalent of our OD reading. So if we have that extinction coefficient, we can get the concentration of our sample. So the slope is now directly proportionate to the concentration. This is critical in any slope spectroscopy method that we're using, whether it's proteins, plasmids, color analysis, or polysorbate. The rule is the same. If you're scanning at the wavelength of interest, the slope becomes your new OD value in any measurement going forward. And really all the benefits that we see with Solo that are listed on the right here, a lot of them, most of them are really related to dilution, the time-consuming process that it has, and the error-prone variability that it introduces. So this is our brand new Viper Analytics software that we're going to be showcasing right now. So this is an extremely easy and intuitive software. It's 21 CFR compliant, and the most important thing is it's SQL Server database driven. So we've completely gotten rid of the old UV platform that we had, and we're now completely independent on our own software. And all the data that is generated will be saved into a SQL Server database file. And now we've provided a lot of customability so that you can actually configure your system to be 21 CFR compliant or however compliant your group is. So you can really lock down your system and ensure that data integrity is achieved. We have a whole team of bioanalytics application specialists for each system that's purchased to ensure that implementation of the system goes smoothly. So now that we covered some of the basics on slope spectroscopy and the software, we're going to get into applications for plasmid manufacturing with a few main examples. So the main areas of interest here are culture density, plasmid purity, oligonucleotide determination, and titer determination. So let's start by talking about the OD600 reading for culture density in cell harvest and E. coli fermentation. So as most of you know, we're learning to monitor cell growth over time. We're not changing anything from the previous method here. The wavelength and techniques are identical, and we're just replacing single absorbance readings with slope regression. So using slope spectroscopy gives us an advantage because as cells grow, the solution they're immersed in becomes more and more concentrated. For these readings, dilution's not required, so with the SoloVPE, right away, we're going to eliminate any errors associated with dilutions. And we're also going to eliminate a lot of the issues related to time. And by that, I mean, when samples are pulled, there's still cell growth within a given time point aliquot. So since we won't be diluting now, we can actually measure the sample immediately. And by gaining that time back, we are actually much more accurate to that target value. So, now is a great time to mention that, not only do we have the SoloVPE, but we have a FlowVPE that can monitor cell growth in real time because the FlowVPE can actually be connected in process. But for this purpose, we're mainly going to be talking about our at-line SoloVPE device. So all of this data was generated by the BCC in Switzerland, and essentially they were trying to prove that the UV technique was the same as the Solo, and that they would get the same value. So this sampling was done every 30 minutes, and you can see that with traditional UV, half of this process required dilution, and with the Solo, again, no dilution, real-time measurements, and no difference in the method, other than replacing that single absorbance reading with a slope reading. So this slide covers antisense oligonucleotides, or commonly known as ASOs. So these have become more and more important in the drug discovery platform. Currently, there's only 6 approved drugs, but there's actually over more than 50 in clinical trials. So this is certainly a sign that this is the future of gene therapy products. So during different operations in manufacturing, in-process controls must be implemented to ensure that ASO assay results are within acceptable range. So from all of this work on this publication, we were actually able to determine that SoloVPE, had a much simpler instrument setup, test procedure, and data analysis because all the data analysis is completely automated. We definitely saw increased turnaround time, reduced time to market, and increased throughput. So it was actually determined that 67% of time savings was achieved with the SoloVPE for ASOs because the HPLC method actually takes 6 hours. So this is just another chart of the data that was generated. So we were actually able to determine that SoloVPE certainly is the most efficient and universal IPC assay instrument and method for ASO drug product manufacturing. And that's because linearity, repeatability, intermediate precision, robustness, and reproducibility all were an improvement with the SoloVPE. So all the other reasons why all of our other applications see a benefit for the SoloVPE is the same here. Mainly, most of our data analysis is automated or simplified, and the lack of dilution doesn't introduce any additional variables for error. So one of the devices that was compared to the SoloVPE is the NanoDrop, and it's understandable why some labs use the NanoDrop, low sample volume. However, it still has the same issues as a traditional UV instrument in that it's still a fixed path length technique. So if the sample's beyond the linear range of the NanoDrop path length, then dilution is required. So this poster here was presented by a company that worked with us called Amgen, showing their transfer of the nanodrop-based nucleic acid studies into making the SoloVPE their primary platform method. Like most pharmaceuticals, concentrations are increasing. When they stayed at 3 mg per mil, the concentration was comparable. But as soon as they increased the concentration, the SoloVPE was able to handle it, and the NanoDrop was no longer able to, and they were forced to introduce dilution steps. So just to mention, being able to provide those same consistent repeatability at plus or minus 2%, Amgen actually carried over from their protein work into nucleic acid. So they've actually established SoloVPE as their main platform measurement for nucleic acids. And this is another publication for SoloVPE with DNA purity, nucleic acids. So this study highlights another company that we worked with that now has established SoloVPE as their platform measurement through a lot of sample testing. And they actually had samples sent in from other labs of 25 different sample concentrations and compared the results in relation to their theoretical target value. So the company we worked with actually used their own internal product, but for this publication, they actually got independent sources and both labs ran that material as a blind test. So same as the other poster that I just showed, all of the results had a percent difference below 2% of the target. So this publication also goes into detail on how these methods were set up. So now we're going to talk about customer success. This is a term we really like using at C Technologies because one of the things that we really like to separate ourselves from our competitors is our customer interactions. So on this next slide, I'm certainly going to talk about some of that here. So upon every purchase of the SoloVPE, technician comes on site, installs the system, they provide a training, they schedule a time beforehand, and then one of our application specialists like me will reach out to the customer to ensure that system implementation goes as smooth as possible. And we've actually created several documents for this. So we have a computer system validation document that the customer can completely validate their computer with. We have SOP protocols that we will actually edit SOPs completely for free of charge. And all of this is just to ensure our customers' success in implementation into the lab. Especially in the 21 CFR compliance, we understand that this is a timely process. There's a lot of learning about new instruments and how they can be set up to be 21 CFR compliant. But it's just nice to know that with the SoloVPE, you're not left alone after purchase. We are completely with you through that entire process, just to ensure that your success actually translates into our success. So we're certainly motivated to help out. And now it's actually important to mention that when Repligen purchased us, we actually gained the ability to become a global support network. So no matter where you're located in the world, we are actually very within reach because we have a global support network in Europe, Asia, and North America. So just to reiterate with the SoloVPE, you're going to reduce time and variability. All of this is related to the fact that now we're not diluting. We got 10 data points now. We have a linear check for every single measurement. So every single time the Solo has been used, we've had a high level of repeatability within 2%. And the process certainly is optimized because of the time that's being saved, not spending time diluting or analyzing the data afterward. So with the SoloVPE, we're certainly going to save you some time and labor. That's no different than any other application with the SoloVPE. The UV technology we have as the widest concentration range with direct measurement. Our path length resolution is unique to the Solo. There's no other UV technology able to take such small and precise path length steps to generate linear data for a particular sample. It's reproducible. Plus or minus 2% claim has been in place since 2008 on the original spec sheet and hasn't changed since. And the transfer of methods has become drastically more simplified by eliminating the most error-prone steps of the process. So again, being part of the Repligen family gives us this global support network. So we're able to reach our customers all around the world. And you can certainly ask any more information that you'd like by clicking the link below, or you can email me at my email listed here, understanding that slope spectroscopy isn't in our universities right now. So the only way that scientists have expertise is with actual experience. So we're certainly open to people inquiring with questions about the technology. So I highly recommend that. Thanks everybody for attending. Thanks for your time. And I hope you consider SoloVPE in your future analytical process. Thank you.
Thank you, Paul, for a great presentation. And thank you for watching today. If you'd like to download a poster version of today's talk summarising the main points, you can find that below the video. We hope you'll join us again soon for more fast facts.